Thank you, REPROCELL, for the opportunity to be here presenting today. I'm very excited to tell you a little bit more about Gameto, but also about Fertilo, that, as Bob mentioned, is our iPS-derived ovarian support cell as a solution for infertility treatment. So in these slides, you just have my disclosure, but most importantly, I would like to acknowledge this amazing team of scientists and the team overall who have been working day and night to make all this work possible. So Gameto's mission is to create a portfolio of treatment solutions to support women throughout their reproductive journeys. So this includes developing treatments for medical problems caused by ovarian decline. And as some examples, we have this goes just from since from egg freezing, when it allows the woman to have more control about their timelines, their reproductive timelines, but also in vitro fertilization or IVF, up to menopause solutions when the ovarian function is already declined and lots of women suffers with consequences of that. So at Gameto, we work with a platform of ovarian support cells that are derived from induced pluripotent stem cells. This technology was developed initially at the lab of George Church at Harvard, and it relies on expression of specific transcription factors to drive the differentiation of the iPS cells into cells of the female reproductive system on a dish. With that platform, we have multiple assets. And in my talk today, I'll be focusing on Fertilo, which is our lead asset. And we are very excited about all the progress in this work. So Fertilo is a highly differentiated solution with potential to disrupt the IVF and egg freezing market. Right now, it's estimated that just in the United States, about 9 million women may be experiencing fertility. Despite this huge number, just about 300,000 artificial reproductive technology cycles happen per year. And just about 50,000 babies, live births from these cycles happen every year. So this gap between the amount of women experiencing fertility and the amount of babies being born per year through this technology is believed to be due to limited access to those technologies and also some efficacy related to these processes. Therefore, there's a huge need for expanded access and for further development and innovation in this market. And despite these limitations that I just mentioned, there is increasing interest of the society into using these procedures for multiple reasons. And some of those, the drivers of those increasing interests are, for example, cultural shifts. So women have been delaying more and more motherhood, or same-sex couples interested in having their own biological kids through these technologies. In addition to that, there is an increase in chronic health conditions that are driving also an increase in fertility rates, and more awareness and accessibility. For example, health insurance plans supporting reimbursement and adding these treatments as part of their benefits. So let me walk you through how this process usually works in most clinics to these days. So both for egg freezing or for in vitro fertilization, the entire process is very long, very painful, and very costly to the patient. So usually a woman will ovulate about 1 mature egg per month. And in order to have enough mature eggs to allow egg freezing or IVF, we have to trick the body into maturing several eggs at the same time. The way this happens is by inducing tons of hormone stimulation. This usually happens with hormonal injections, about 2 injections a day, in a process, in a cycle that takes about 2 weeks. To give you an idea, the average to lead to a pregnancy is about 3 cycles. So now we are talking about 6 weeks of this 2 hormonal injections per day in order to have a pregnancy. And all of this is also accompanied, the patient has to have constant medical monitoring in order to make sure everything is going well and to avoid severe consequences. So during this process, 1 in 3 women can experience ovarian hyperstimulation, which is a painful and potentially life-threatening response to this stimulation. This process is also extremely expensive, not just for the cost of the hormones itself, but all the medical monitoring that it's required also adds to the cost. And despite the ovarian hyperstimulation that I mentioned, there is a series of other side effects such as mood swings, headaches, anxiety, depression that can be associated with this stimulation process. So what would be an alternative, a solution to make this process a better experience for the patient? One possible solution is known as in vitro maturation, or IVM. And in vitro maturation relies on the capability of maturing these eggs outside of the body instead of triggering the body to mature these eggs artificially. In vitro maturation has been developed since 1991. And to this day, it's already considered by the American Society of Reproductive Medicine no longer an experimental technology. In vitro maturation makes the whole process faster, safer for the patient, less painful because now we are talking about no injections or very few injections compared to the standard cycle, and a lot more accessible. So there's no disruption on people's lives. And because it's cheaper, it also allows more people to be able to do it. The problem with in vitro maturation is that it's still not as efficient as in vitro fertilization and the methods used for egg freezing, and that's why it's not more widely adopted. So what Gamita wants to propose with Fertilo is to have this new platform to make in vitro maturation much better, and in consequence provides a better experience for the patient, a safer and more accessible alternative to IVF and egg freezing. Instead of relying on the culture media to support maturation of this egg outside of the body, now, with our ovarian support cells differentiated from the iPS cells, we can provide this ovarian environment on a dish to support this orchestrated final maturation of the egg. And what Fertilo is doing on a dish is Fertilo is talking to the oocytes, but most importantly, the cells that come attached to the oocytes that are known as cumulus cells, and they are the ones that are actually supporting the oocytes to finalize this maturation process. So this conversation allows the maturation happens on a more reliable way than current or more orchestrated way than in the current methods for IVM. And also because these ovarian support cells that are being provided with Fertilo are derived from stem cells, they also have a younger phenotype than what it would be in the ovary of most women interested in moving forward with these procedures. So Fertilo increased the rate of human oocyte maturation in vitro and improved quality of blastocysts. Let's go through the proof of concept. As I mentioned in the beginning of this talk, this technology was developed in the lab of George Church at Harvard by Christian Krem, our CSO. And in the seminal work, Christian and colleagues, they were able to screen several transcription factors to come up with the minimal combination that would efficiently differentiate the pluripotent stem cells into these ovarian support cells. This combination of transcription factors allow that the pluripotent stem cells turn into this, what we call ovarian support cells, that not only provide and support the maturation of the eggs outside of the body, but they also have this ability of organizing themselves into follicle-like structure and functionally in a dish very similar to follicles in the human body. So in this study, I'm showing to you one of our first proof of principle in which we use this differentiated cells, these ovarian support cells, to mature oocytes outside of the body in parallel with the standard protocol for in vitro maturation. What you see in green in the graphs that I'm displaying are the results for the standard protocol of in vitro maturation. And what you see in purple are the cells, the oocytes that were matured with supplementation of Fertilo. We see that Fertilo not only improved the maturation rate of the oocytes compared to the traditional method, but it also improves the percentage of euploid blastocyst in comparison with the traditional methods, suggesting that this maturation is leading to high-quality eggs that are also leading to high-quality embryos. At Gamito, we have also done several non-clinical studies to demonstrate Fertilo's efficacy, but also to demonstrate that there were no behavioral or developmental abnormalities associated with the progeny and the next generations of this mice. I'm not going to get super deep into this work today, but you can find this data on our publications online. So the goal of the talk today, we're going to walk you through some of the initial changes that we did on the protocol of differentiation of the ovarian support cells to translate a protocol that was initially developed in a research lab towards a protocol that can now be used for clinical and commercial manufacturing. So the process of differentiation, manufacturing of Fertilo, it's an extremely straightforward and fast process. We can bring the cells from the iPS state to the ovarian support cells in up to 10 days. So our protocol consists of an initial 5 days of iPS cell expansion, followed by 5 days of differentiation, which again, it's driven by the overexpression of those transcription factors, as I mentioned. In this flow assay that I'm showing to you, you see the expression of CD82, which is one of the main markers of ovarian support cells. And we see that after this 10-day process, most of our cells are expressing that marker, showing the efficiency of this protocol. In order to make this protocol into a product, one thing that we have to carefully do is to really understand what are the cells and what is the signature of the cells we are generating. So for that, we used single-cell RNA sequencing experiments in multiple batches of differentiation to further characterize this different cells and different subtypes of cells that we were seeing. What we can see in this plot, the single-cell RNA sequencing plot, is that most of our cells acquire a phenotype that we call GC-like, granulosa cell-like. And from this, the cells that are acquiring this phenotype, we have a subpopulation of cells that have a phenotype a little bit less mature that we are calling early GCs, and cells that have a phenotype that are a little bit more mature that we are calling GCs. There's also a small subgroup of cells that have enriched a mitochondrial ribosomal gene expression. This is usually common to single-cell RNA sequencing data, and it can be associated with some artifact of the technique. But because cell death is also part of the process and part of the biology of those cells, we classify these 3 subclusters as others, as we were not able to identify if they were just cells undergoing the biology or artifact. But the cells we're going to be focusing are the ones that have the phenotype of the granulosa-like cells, both the early GCs and the GCs. We then differentiate 6 independent batches of cells that had differentiated cells that had undergone this protocol using still the research conditions. And what we were able to notice is that there was some variability from lot to lot. So I just mentioned to you that the protocol is very efficient in generating cells with the granulosa cell phenotype, but the subpopulation, the early GCs and the GCs, were not very consistent among these lots. And this can pose a limitation for us to turn this into a consistent product. So as part of the process of optimizing the protocol, we did change several reagents to raw materials and reagents to use now reagents that are GMP grade or of the highest quality available. And one of the most important reagents that we had to change throughout this process was our substrate. So in the research protocol, we were using a common substrate that is usually extracted from a mouse tumor. So this reagent has the issue of being animal derived, but also it carries the burden of being very dependent from lot to lot as it is an extract of a tumor. So we had, in the process of selecting what substrates we were going to substitute this one for, we decided to test, among others, vitronectin and laminin, which are common substrates used for culture of human pluripotent stem cells. So we differentiated our stem cells onto vitronectin or onto laminin. And we were able to see that after the entire differentiation process, in both cases, most of the cells are expressing CD82, showing that both substrates are efficient in supporting this differentiation towards granulosa-like cells. However, when we take a careful look through single-cell RNA sequencing, we see that the cells differentiated onto vitronectin, they have a profile that it's closer to what I showed you before of the early granulosa-like cells. They seem to be a little bit less mature, and the cells differentiated onto laminin, they seem to be more concentrated into that more mature phenotype of granulosa-like cells, and we have barely no cells of that other state that we called others. When we look at individual lots, individual batches of cells differentiated in either of those two substrates, we see that now the change of the raw materials, including the substrate, really support a much more reliable differentiation in each of the cases. So we see this increasing reproducibility in both of them, but in particularly on the laminin. Another big thing that we had to change, in addition to all raw materials and other details in the process, was our starting material. The cell line, we did some of the initial data that I showed to you was a cell line that is a research grade cell line. So this involve several protocols, including consent. And we had to bring this to clinic and commercial, we had to source a female cell line that was also clinical and commercial grade. So this seems there is now more and more new therapies coming up using induced pluripotent stem cells as a starting material. But it is interesting to think that most of the starting material available, especially the ones off the shelf, they are usually from male donors. So we had some challenges to find some female donors, but we were able to source one cell line from REPROCELL. We tested two female lines, and we are moving forward with one of them that is generated in GMP using mRNA, so really not changing the genome to allow this reprogramming, and had been further characterized by REPROCELL and then by us. So after identifying that cell line, we had to create the same changes, the same gene editings that we had in our research cell line to allow these cells to have the inducible transcription factors that would drive the differentiation into the ovarian support cells. So we started with this process. We engineered the cells to insert our three transcription factors. We screened multiple clones. And here I'm showing to you a pool of final clones that had the insertion of the three transcription factors, were growing in good rates, and we decided to further characterize to select our lead clone. So in this process, we differentiate each of these individual clones into ovarian support cells. And we were able to see that all of those clones had started expressing both FOXL2 and CD82, two markers of ovarian support cells, and expression of OCT4, a marker of iPS cells, was basically eliminated by the end of this protocol, suggesting the efficiency of the differentiation. We also wanted to assess function of the cells. And for that, we challenged the individual clones with FSH and androstenedione to see production of estradiol by these ovarian support cells. After all this characterization, we were able to select a clone that it was our lead clone to move forward with these studies and show further functionality and characterization. So we selected a clone with high viability, high expression of FOXL2 and CD82, low expression of OCT4, and high production of estradiol. We then, when this clone was ready, expanded, characterized, we were able to then perform the differentiation of the cells using exactly the same protocol we had identified and modified for the research grade cell line. We were able to show that differentiated cells expressed high levels of FOXL2 and CD82, and no expression of OCT4 and NANOG, which are the two markers of stem cells, iPS cells, showing barely no residual iPS cells in the final product. We also performed further characterization using single-cell RNA sequencing. And we see that similarly to what we saw with the research grade line using laminin, differentiation of this new clinical grade line led to the same subpopulation of mature ovarian support cells, and that reproducibility was well seen when we look at independent lots. So it's important to highlight that the robustness and efficiency of this protocol is captured not only from lot to lot, but also among different genetic backgrounds. So then our next very important step was to show that these cells are functioning. So for TILO, we know that it acts outside of the body, and we started identifying that this mechanism of action seems to be modulated by interactions with the egg, and most importantly, the cells that come outside of the egg that we call cumulus, and those are the COCs. So we performed similar experiments as I showed to you in the beginning. We had our control, which is the standard in vitro maturation protocol, and we had our samples in which we supplement this culture with the ovarian support cells. We see that compared to the controls that now are depicted in gray, the maturation in the presence and with the supplementation of the ovarian support cells lead to higher rates of M2 cells, which are the mature oocytes. But very excitingly, when we look at independent lots, we see that the rate of maturation is very consistent. So this is good because it shows that that consistent that we are seeing on the cell characterization is also captured when we look at functionality of these cells. In trying to understand how these cells are working, we perform a series of experiments that can be seen in the papers published by our team that include both proteomics, transcriptomics. So we assessed the proteomics of the ovarian support cells at 0 hours and at 24 hours. And from the proteins that were being expressed during this time, we were able to further characterize them and classify them as proteins that are associated, for example, with several metabolic processes. On a different angle, we also did transcriptomics assay on single oocytes. And in this experiment, we were able to look at oocytes that had been retrieved straight from a conventional cycle of IVF, but also oocytes that were matured in vitro with supplementation of fertilo in purple or without supplementation of fertilo following the standard in vitro maturation protocol in green. What we can see is that the changes that we are seeing, the profiles that we are identifying in these oocytes, they are more similar with the supplementation of fertilo with the eggs retrieved from standard IVF. And interestingly, lots of the genes identified there, they can also be classified in function groups that are associated with metabolic processes, suggesting that changes in the metabolism, both from the oocyte as well as from the OSCs, might be related with this process of maturation. And one very interesting experiment that we performed, you can also see this in our publication, we looked at expression of some genes that are associated with the maturation process at 0 hours in these OSCs. We also look how these genes were expressed on the OSCs after 24 hours in culture. We saw some differences that might be highlighting some of the factors that are being expressed by the cells and should be expected with cells being cultured. But the most interesting data came when we looked at the expression of the same genes when these OSCs are cultured in the presence of the oocyte. So now we see that the way that some of those factors are modulated is different. So basically what I'm telling you is that the oocyte is directing the cumulus oocyte complex to lead to orchestrated maturation of the egg, but the egg and the cumulus cells are also, they also seem to be telling our cells, the ovarian support cells, what is needed. So it's a bidirectional conversation in which we believe that the egg and the cumulus cells are requesting, let's informally say, to our cells what they need, and our cells are producing. This dynamic environment is what we believe are supporting the precise maturation of these oocytes compared with the traditional method that it's a culture media and does not allow for that. Finally, I'll just end showing you some new data from some clinical outcomes of fertilo, in which we have already reported some pregnancies, being the most advanced pregnancy successfully already in the 3rd trimester. So we're very excited to see that this technology is supporting the process of maturation of the eggs, but also leading to safe pregnancies. And to finalize, I mentioned in the beginning that our platform was used for multiple assets. In today's talk, I expanded more on our manufacturing process and our advance on fertilo, which is focusing on egg freezing and in vitro maturation. But Gamito has been also very interested in developing, in further developing our other assets, in particular, our asset for menopause, which is closer to a more traditional cell therapy. So the idea is that when the ovarian function declines due to menopause, ovarian insufficiency, the cells from the ovary can be replaced by ovarian support cells and reestablish that conversation in between the ovaries and the brain to reduce some of the adverse effects from menopause, for example, and hopefully also some underlying conditions that are usually associated to menopause. There are several diseases that start after the beginning of menopause, And we hope that we believe that this therapy could also support reducing those. With that, I'm going to end my talk, opening for questions, but I also wanted to put both my contact and of our CSO, Christian Krame, if you want to continue this conversation in the future. So thank you again, Bob and REPROCELL. It's been a pleasure to talk to you today about Fertilo and Gamito.